Hi guys, welcome to a second session DNA replication. In this last class we have seen the structure of the DNA and now we are directly moving on to how to see how this DNA is being replicated. So key points of this session are we are going to see this session as two halves. First is the components of the replication machinery, what is their function, what are their roles, etc. We'll discuss first, and then we'll move on to the mechanism of replication, where we have mainly the three steps: the initiation step, the elongation, and the termination. So we'll be seeing each part as shown in the key points. And at last we'll discuss about the difference between eukaryotic and prokaryotic replication. So if a question is asked about replication, the replication in your syllabus or what we are going to discuss is common for both eukaryotic and prokaryotic. The main difference is regarding the proteins which take part in this particular process of replication. The rest, the mechanism, etc. remains the same, and the differences or the major differences between prokaryotic and eukaryotic will be discussed at the end. So directly moving on to our topic without any delay, we'll start with the first in the key points, which is the components of the replication machinery. So coming to the components of the replication machinery for DNA synthesis, it requires mainly the deoxyribonucleotide in the form of triphosphates and a primer template junction. So we'll discuss a primer template junction first and we'll move on to the other one. So coming to the primer template junction, so as you see in the diagram here, this diagram clearly depicts something which is called as this junction, which is the primer template junction. So in this case you can see that a primer template junction is nothing but here in this diagram you can see this particular portion is what is called as the template, and this one and the one which is seen towards the top is the primer, which is synthesized by the primase, which will be dealt with later. So here you can see a junction. A junction means here there is a junction between the template as well as the primer, and this is a junction where you can see a double-stranded region and a single-stranded region. Double-stranded region is this particular region where the primer is complementary, or primer has already formed hydrogen bond with the complementary bases, and the rest region is a single-stranded DNA. So a primer template junction is a single-stranded double-stranded junction, and this provides, or this particular area is the region which provides the starting point for DNA replication. So the primer, and the major thing is that as you can see in this diagram, the primer, after the primer is being added, you can see an exposed 3' region can be seen here. So this particular exposed 3' region is mainly important for DNA replication to occur. The primer must have an exposed 3' hydroxyl group adjacent to the single stranded region of the template, and the primer is the substrate, or the primer is the substrate region for DNA Pol 3. 3 is not visible here, so make it as Pol 3. So DNA synthesis is DNA is synthesized by extending the 3' end of the primer. Coming to the second one, that is the nucleotides. That is the second component of the DNA synthesizing machinery. So you need 4 deoxyribonucleotide triphosphates in the form of they are required in the form of triphosphate, because while adding, as you see in this particular diagram, while adding, so they are added in the form of triphosphate. One phosphate will be bonded here. Okay, as we'll see here, you can see here the particular so here you can see the primer here, and a new base is being added, and it is in the form of triphosphate, where you can see 3 P's being attached to this. Of this, two phosphate will be removed and one phosphate is being added here for the formation of the phosphodiester bond. So that's why we require phospho or deoxyribonucleotide triphosphates for DNA synthesis to occur. So all the three deoxyribonucleoside triphosphates are required here, that is GTP, CTP, ATP as well as thymine triphosphates. After this, the third component that is required for DNA synthesis or replication is the enzymes. So we are going to see a set of enzymes. So the set of enzymes, the main enzymes that is required for DNA replication is the DNA polymerases. And in E. coli or in prokaryotes, you have 5 DNA polymerases, which is numbered in Roman letters 1 to 5, of which 1 to 3 or only 1 and 3 are involved in DNA replication. Pole 1 is also called as Kornberg enzyme. So please note this. This can be asked as a 1-mark question, Kornberg enzyme. So don't confuse it. Pole 1 is the Kornberg enzyme. And the main function of Kornberg enzyme or pole 1 is the RNA primer removal and DNA repair. So this can be discussed in the later mechanism, what is RNA primer removal, etc. So the function of pole 1 is to remove the primer and the other one is in DNA repair. Pole 1 has it as a single subunit or a single or a monomeric enzyme which is having 3 catalytic site. One is for DNA synthesis in the as normal synthesis in 5' to 3' direction, and it is having 2 exonuclease activity, in the sense that exonuclease means it can cleave off the phosphodiester bond at the ends of a DNA. We have studied about endonuclease in biotechnology. This is exonuclease. Exonuclease means it will be cleaving the phosphodiester bond at the ends of a DNA fragment. So this particular pole 1 is having exonuclease activity in both the directions, that is it can cleave the DNA at both the strands, that is from 3' to 5' as well as 5' to 3'. The problem with pole 1 is that it can synthesize only short stretch of DNA. So this is not the main enzyme that is involved in DNA replication. The main enzyme that is involved in DNA replication in the case of prokaryotes is pole 3. Pole 3, as you see in the diagram, it is a dimeric enzyme. Dimer in the sense that you can see every subunits in 2 units. You can see the polymerase or the core polymerase, etc. You can see the beta clamp, etc. Everything here, it is in the form of it is duplicated. That's why it is called as a dimer. The pole 3 is a primary enzyme which is involved in replication. So pole 3 is having the normal as a polymerase. It is having 5' to 3' polymerase activity, and it is also having, it has only 3' to 5' exonuclease activity. It does not have 2 exonuclease activity as with pole 1. It's having only 1 exonuclease activity. The next is coming to the components of this particular enzyme. As we said, it is a dimer, and the holoenzyme consists of 2 core polymerases. These are the 2 core polymerases which is here, and it has something which is called as a beta clamp. So the core polymerases means you can see this particular core polymerase and this particular region and beta clamp. So these 3 together forms what is called as a holoenzyme. So the function of this beta clamp, as you see, can see, it is a C-shaped clamp like structure. Its function is, so in this diagram itself you can see that along with the core polymerase a beta clamp is there in the form of a ring, so which is marked here. So that is a beta clamp. So the function of this beta clamp is to allow the polymerase to remain associated with the DNA. As this is the main enzyme that is involved in DNA replication in prokaryotes, this can synthesize long stretches of DNA when compared to pole 1. I think there is some mismatch which is given in your textbook regarding the Kornberg enzymes, etc. So please note that pole 1 is Kornberg enzyme, POL1 is an enzyme which helps in replication. We'll see what it does in replication, but that is not the main enzyme that is involved in replication. It is POL3. I think that is clear. And mind you, POL1 is a monomer which is having 3 catalytic sites, and POL3 is a dimer which is having mainly 2 functions. One is polymerase and the other one is a single or a unidirectional exonuclease activity. So coming to the next enzyme, so after that we have completed the set which is called as polymerases. Next is ligases. So after the POL1, so we'll see what is an RNA primer, etc. So after POL1 replaces, we said that the function of RNA POL1 is to replace a primer. After the primer is being replaced, RNA primer is being replaced, and because of its polymerase activity, it synthesizes or it replaces the RNA to DNA, and there will be a gap between the primer region and the rest of the region. So the formation of the phosphodiester bond is being catalyzed by this particular DNA ligases. Next is the primases. Primases, it is an RNA polymerase which synthesizes short stretches of RNA, which is usually lesser than 10 nucleotides, which is complementary to the template. So a primase is an RNA polymerase which can synthesize short RNA fragments, which is also important in the mechanism of replication, which we are going to see. The next category of enzyme is a helicase. Helicase moves along the DNA. It uses the ATP and it unwinds the duplex. So unwinding the DNA or uncoiling the DNA is a function of the helicase. The next category of enzyme is topoisomerase. So please note the name gyrase. Gyrase is that topoisomerase which is seen in prokaryotes or mainly in E. coli. So please note the term gyrase, which can also be asked for a 1-mark question. Gyrase, it is a topoisomerase. So all topoisomerases and gyrases, it removes supercoils ahead of the replication forks. That the same thing we'll discuss later in the coming sessions. So these are the set of enzymes that is involved in DNA replication. Next is the protein. So there is only one major protein which we will be discussing in this particular topic, that is a single stranded binding proteins or SSB. So as the helicases unwinds the DNA, so there are chances that the complementary strands, they again forms the hydrogen bonds, come together. So our intention is they should be separated and a new strand should be synthesized for both. So for that to happen, these proteins bind to these DNA and they allow them to be separated, or they prevent them from reforming the duplex. And this particular the proteins are tetrameric proteins. That means they are having 4 subunits in it. And before ending this particular session, you can see, or you should be noted with 2 terms which can also be asked for 1-mark question. One is a replisome and the other one is a replicon. So replisome is nothing but it is often referred to a complex of proteins that is active during replication. So active during replication, in the sense, a replisome means it is a particular complex which consists of the DNA polymerase holoenzyme, the helicase, the SSBs and the primase. So these 4 together, or this complex is called as, this particular complex is referred to as the replisome. Next one is a replicon. A replicon is a unit of DNA which is engaged in replication. That is, which part of the DNA which is engaged in replication is called as a replicon, which is the region where replication bubble or the replication eye is seen. So with this, I am just ending this particular video because the size will be higher. So we'll see the mechanism of replication in the next video. So in this video, we have covered the components which are involved, their function, etc. In replication.